Okay, so good day, Clin Pharm 114 students. So this will serve as your supplementary lecture for exercise 4 staining techniques. So because remember during our pre-lab discussion for exercise 4, we tried to condense it to make it shorter so that you will have more time to perform your actual experiment. And so we'll just go through the other important concepts that we were not able to go through anymore during our pre-lab discussion. Okay, so let's just review the objectives of our lecture. So this time around, we'll be able to describe the types of staining and their importance in more detail, and also differentiate the types of stains, and also will give emphasis on the differences in the cell wall and also the different structures of your gram positive and gram negative bacteria that contributes to the staining technique that we know, the gram staining technique. And then you were able to perform already your proper staining techniques when we did your experiment. So again, why is it important that we perform staining when we try to observe the bacteria under the microscope? So because naturally your bacteria would be colorless, meaning they do not have any color, so you will not be able to properly observe them under the microscope. Yeah. So they will just look similar to this one sa so what you see in the picture. So they're just parang colorless na mga cells under the microscope. And then when we finally place suitable dye or stains, they would now look like this, na meron na silang color. So you'd be able to appreciate them better and visualize the structures better as well. Okay. So actually, we can categorize or have different ways in order to differentiate our staining techniques. So you encounter the different staining techniques in your laboratory manual. So the first one of which is your simple staining. So your simple staining, so basically, again, we mentioned that you only use one type of dye. It's either you use an acidic or a basic dye. So again, single dye in contrast to your other staining techniques wherein you use more than one dye or stain. So di ba merong primary stain and then meron pang counter stain and other reagents as well such as your mordant and also your decolorizers. Yan. So, but when we only employ the use of only one stain, that is your simple staining technique. So this was, we did not perform this anymore for exercise 4, but we were able to perform this during your microscopy exercise wherein you used methylene blue when you tried to view your Lactobacillus casei and your Bacillus clausii na samples for specimens. Another type of staining, so when we say differential staining, so you try to differentiate na, no, different type of bacteria or uh, based on their reaction on the staining technique that you perform. So some examples of this that you already encountered as well are number one, your gram staining technique, and then your acid fast staining. So when you perform your gram staining, you try to distinguish between gram positive and gram negative bacteria. Pag naman sa acid fast staining, you distinguish whether the bacteria is an acid fast bacteria or a non-acid fast bacteria. Yan. So lastly, we also have your miscellaneous staining techniques. So this time around naman, we try to identify the specific structures of your bacteria. So this time around, you try to focus on staining particular structures in the bacteria. So when you try to visualize yung capsule, so that is capsule staining. Kapag naman you want to look at the spores, spore staining. And then also, if you want to look at yung flagella of your bacteria, so that's flagella staining, etc. Okay. So another way in that we can also categorize your stains. So this time around, yung stains naman itself that we use, we can categorize them to be acidic, basic, or neutral. Okay, so the stains that we have in the laboratory, in the micro lab, um, they are actually composed of salts. Yan. So when we say salt, meaning it has a positively charged and a negatively charged ion or component. Okay, so uh, the 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 category of your stains will actually depend on whether the chromogen is positively charged or negatively charged. Okay, so your stains, it is also composed of your parang organic portion of the dye that is responsible for the coloration. So that is what we call the chromogen. Okay, so the dye is composed of your chromogen and another ion that is attached to it. Ngayon, the what will determine kung acidic ba siya or basic is the chromogen. When the chromogen is a negatively charged na compound, yan. So we say that the stain is an acidic stain. Okay? Kasi later we'll look at the structure of your bacterial cell and we'll see na actually generally your bacterial cells are negatively charged. So if your chromogen is negatively charged, naturally it will repel the cells that are generally negatively charged as well. Yan. So that's why we say na acidic siya kasi negatively charged yung kanyang chromogen component. Yan. So meaning it has H plus ions. On the other hand, kapag naman the chromogen is positively charged, excuse me, kapag positively charged yung chromogen, so we say that the stain is basic. Okay? So some examples of your acidic stains are your nigrosin, eosin, and Indian ink actually is another term for nigrosin. Yan, picric acid. For basic naman, some examples of stains that we know are your methylene blue, safranin, crystal violet, malachite green, carbolphoshine. Tapos lastly, we have your neutral stains. So this one naman is a combination of both your acidic and basic stains. Okay? So usually they are in the form in the aqueous form. So one example of this is your GEMSA stain. So for us to just visualize better, no, ano ba yung characteristics of your bacterial cell membrane that contributes to the negative charge, overall charge of the cell. So as you can see, regardless if your bacterial cell is gram positive or negative, generally it is negatively charged. So bakit kaya? As you can see here, for your gram positive bacteria, there is the presence of compounds or mga strands here in the form of your lipoteichoic acid and teichoic acid. So these compounds are actually negatively charged, no? So it contributes to the general negative charge that is found in your cell membrane. At the same time, the peptidoglycan layer present both in your gram positive and gram negative bacteria, although mas makapal lang pag sa gram positive. It is also composed of your carboxyl and hydroxyl ions. Yan. So they also contribute to the, carboxyl and amino groups in the peptidoglycan layer. So it also contributes to the negative charge. Sa gram negative naman, so the presence of your lipopolysaccharides as well contributes to the overall negative charge of your cell surface. Yeah. So the presence of your saccharides, your sugars, complex sugars, would contribute to the negative charge. And so we saw na the cells of your bacteria are generally negatively charged. So yung mga acidic stains natin containing a chromogen that is negatively charged, naturally it will repel the cells of your bacteria. So meaning your acidic stains would now will not be able to stain your cells themselves. So that's why these are the stains that we use for negative staining techniques. And so because your acidic stains would be used to color the background and not the cells themselves. On the other hand, yung basic stains that are positively charged, yung chromogen nila, so meaning the chromogen would be able to attach to the bacterial cells na negatively charged kasi na positively charged sila, so they would attract. Now, your basic stains now can colorize your bacterial cells. So let's just look at some examples or some pictures of what your acidic stains and basic stains would look like. So this is just a picture of a negative staining technique wherein possibly nigrosin ang ginamit dito because black yung kanyang background. And as you can see, the bacterial cells were not stained because the bacterial cells were not able to absorb the chromogen. Yan. So that's why it remains colorless. Similar to these other pictures wherein, as you can see, ang na-colorize natin is the background and not the cells themselves. So this one naman are some examples of pictures wherein we used basic stains. So ito, possibly, since it's blue in color, baka methylene blue yung ginamit dito. Ayan. As you can see, the cells themselves are the ones that are colored. Tapos, the background is colorless. So, for basic stains, the cells themselves will be able to absorb the stains. Okay? So this one naman, this is your spore staining wherein yung spores were able to absorb yung malachite green in the spores. So again, for your neutral naman, an example that we have here is your GEMSA stain. So yung GEMSA stain, so GIEMSA. So you'll also encounter this later when we talk about your parasites. So GEMSA stain is actually a widely used na stain in studying your blood cultures or blood samples or blood smears of patients that have parasitic infections. So some examples would be your malaria. So yung mga Plasmodium species, when they are in your blood, they can be better visualized with the use of your GEMSA stain. So this one is actually an example of a picture that is a blood culture wherein the patient was infected with trypanosoma. So it's another type of parasitic infection. Yeah. Wherein the parasites are being stained with the component of your GEMSA stain that is attracted to the cells of the parasites. Okay, so again, fixing is another step that is important to employ when you perform staining. So, but not all staining techniques would require fixing. So, remember some of the procedures would require na dapat no fixing what is being done for the smear preparation, such as your negative staining technique, and also for your capsule staining. Important na the smear is not subjected to heat in order to preserve the structure or the morphology of the cells. Because the fixing step may actually contribute to the shrinking of the cell. There are changes in the natural morphology. So sometimes fixing is not being employed. But generally, when we perform staining, we first fix the bacteria or fix the bacterial cells on the slide first. So you already know how to do this. So you simply let the smear pass through the flame mga around 3 to 4 times. So the purpose of it is to attach the cells on the slide so that when you wash it off later with water, you would ensure that the bacterial cells will remain on the slide. And also it preserves internal and external structures, inactivates the cell enzymes, so you try to kill the bacteria. So it's also an added protection for you as the experimenter na when you subject the bacteria to heat fixing or chemical fixing, ma-activate yung cells ng bacteria. So essentially, the bacteria is being killed. So it's also extra protection for you. And sometimes these cell enzymes may also disrupt with morphologies. That's why we try to inactivate them as well. So also it toughens cell structure to remain unchanged during the staining process itself and the observation. So there are the fixing step that we know is through the use of heat. Yan. So when we try to pass it through the flame, we perform heat fixing. But actually, another way to do fixing is to employ the use of chemicals. So usually this is being performed with tissue specimen. So that's why the more commonly fixing method that we do is your heat fixing. So some examples of chemicals that we can use for chemical fixing would include your alcohols like your ethanol, methanol, even your aldehydes, mga formaldehyde, butyraldehyde, etc. Yeah. So some advantages of your chemical fixing is that you're able to preserve most cell structures, inhibits chemical reactions, and also stabilizes cell structures. Okay, so now let's discuss the staining techniques themselves. So we already went through this, so we'll just look at the parts that we were not able to discuss anymore. So when we say simple staining again, you use only one stain. So the main purpose of it is to visualize the cells in the smear. Now, when we say simple staining, so yung mga stains that you know, the basic stains that you know, that are also being used with the other staining techniques such as your gram staining, your acid fast staining, your carbon-fuchsin, yan, your safranin from your gram staining or your spore staining. So when they are used alone or they are used as standalone na mag-isa lang siya. So that's when you actually employ simple staining. So meaning what I want to try to say lang is that the stains that you use in the other staining techniques, when they are being used as a standalone stain, you're actually using them as a simple staining technique. Yan. So meaning these different stains would have their own contact time or wait time. So when we say contact time, it is the time it takes for the stain to properly adhere to the bacterial cells. So when you try to use yung mga the different stains for simple staining, you just have to take note of the varying contact time that you need to to make the stain properly adhere with your bacterial cells. Yan. So you just have to review it. Yung crystal violet, usually it's 1 minute. Carbon-fuchsin, mga 15 to 30 seconds. Methylene blue, around 1 to 2 minutes. And then safranin, around 30 seconds. Okay? So, ayan. So, very straightforward lang yung ating steps here. You just have to prepare the smear with fixing, stain the smear. So, remember to take note of the contact time. And then remove the stain with blotting paper. So, carefully lang. And then usually we observe under OIO so that we can properly visualize the cells. So, remember, this is the proper way of how we write yung observations natin under the microscope. You have to include all expected na mga characteristics such as the color, the shape, the arrangement, and even the color of the background. You have to, and if you observe other structures as well, so you have to um, report them as well. Okay, so again, some problems that we encounter when we perform simple staining. So maybe you encountered this when you performed your microscopy exercise, wherein you observe that there is a halo appearing around the stained area. So this happens when you are not able to dry properly your smear. Tapos you applied oil. So, di ba, oil and water will not mix. So, it can actually also contribute to the appearance the of your observation under the microscope. Stain is too light or too dark, so this is associated with your contact time. So kapag masyadong matagal, there's a chance na too dark ang iyong stained cells. Kapag naman too short yung iyong contact time, sometimes it would appear too light, and no cells were found in the smear. It's possible that during blood drying, you wiped it off, or during washing with water, you na wash off from the slide yung smear mo, possibly because the fixing technique was not proper. Smear is too thick or pasty, so when you try to obtain a bacterial growth that is too thick or too much, and pagdating later on sa smear, too thick, so meaning light can no longer pass through the cells, or when you try to observe it under the microscope, they would appear as clumps or clusters when in fact, hindi naman yan yung natural arrangement niya. Diba? So, yeah. Okay, so another staining technique that you encountered, so one of our differential staining techniques is your gram staining. So gram staining is a technique that was developed by a scientist, si Hans Christian Gram. So that's why when you write gram staining or gram positive, gram negative, you don't write it as small letter. So gram with a big letter G, because it is actually named after a scientist, yung last name niya, Gram. Okay, so when we say gram staining, basically what we want to find out is to categorize your bacteria into gram positive and gram negative. So what's the importance ba of doing this? So diba, um, if you remember from your pharmacology, yung mga gram positive and gram negative bacteria, they would actually be more susceptible to different kinds of antibacterial agents. Like for example, your cephalosporins. Cephalosporins that are belonging to the first generation would be more effective against gram-positive bacteria. Pero pagdating natin ng mga third generation na, they would be more effective against your gram-negative bacteria. So when you now figure out kung ano ba yung group ng iyong causative agent ng infection ng isang patient, you would be able to identify the most optimum treatment for that infection, so that the patient will recover more quickly with the identifying the most optimum na treatment for the patient. Okay? So before we go to the actual uh, staining principles behind the colors that we observe with your gram staining, let us just look at the different pertinent characteristics of the gram positive and gram negative bacteria that is actually contributing to your um, contributing to the gram reaction of the bacteria. So let's look at first your gram positive. So actually, the main component of your bacterial cells that contributes to the gram reaction would be the peptidoglycan layer of the cell wall, or the peptidoglycan that is present in the cell wall of your bacteria. Okay, so your gram negative, generally they have your thick peptidoglycan layer. Gram-negative, they also contain peptidoglycan in their cell wall, but it is thinner in so it is thinner for your gram-negative bacteria. Okay? So later we'll see na actually the peptidoglycan layer of your bacteria is the portion that absorbs or that interacts with your crystal violet, the primary stain that is involved in gram staining. Okay? Now, so yun yung main portion of your gram-positive bacteria. So after your thick na peptidoglycan layer, now comes your cytoplasmic membrane. Ito na siya. Ngayon, you would possibly think na yung gram-positive, since they have a parang thick protection, no? Their cell wall has a thick peptidoglycan layer. Therefore, they are more resistant to being attacked by like enzymes or your antibacterials. But actually, it's the other way around. Your gram-negative bacteria would actually be the more virulent types of your bacteria as compared to your gram-positive. So later so let's just discuss bakit kaya ganun. So although yes, it has a relatively thinner na peptidoglycan layer, the gram-negative bacteria also has different components in their outer membrane. Yan. So the outer membrane of the gram-negative bacteria contains what we call your lipopolysaccharides. Specifically, we have here your lipid A. So lipid A, later we'll look at the importance of the lipid A component of the gram-negative outer membrane. Okay? So after nung thin na peptidoglycan layer, now we have your inner cytoplasmic. So again, thicker peptidoglycan, thinner peptidoglycan. So because of the presence of a thicker peptidoglycan layer, so pag sinabi kasi nating peptidoglycan, peptides plus glycans or your mga um, carbohydrates in nature. So, since maraming strands ng mga peptidoglycan, there is a chance that these peptidoglycan strands would interact with each other. Therefore, there is a presence of cross-linking doon sa cell wall. Okay? So, we'll look at dito naman sa gram-negative, since medyo thin lang yung kanyang peptidoglycan, cross-linking is not usually observed and sometimes it is absent. So let's look at the importance of this concept of cross-linking within the peptidoglycan cell wall. So let's just go to sige, balikan lang natin when we discussed earlier the lipid A, which is part of the outer membrane of your gram-negative. So yung lipid A, it actually contributes to the virulence factor of your gram-negative bacteria. So despite having a relatively thinner na cell wall, it contains naman lipid A sa outer membrane, and lipid A is associated with endotoxins present or that is being secreted by your gram-negative bacteria. So these endotoxins, these are parang toxic materials, no, that that contributes to the severe reactions in humans when they are infected with bacteria that are belonging to the gram-negative group. Yan. So yung mga endotoxins na ito, they are the ones that would cause your fever, the lowering of blood pressure. Yan. So meaning, actually when we perform yung mga sterility testing of pharmaceutical products, we try to see if meron bang presence of endotoxins, so that we try to see if there is presence of bacteria, gram-negative bacteria. Yan. So what I'm trying to say is that although medyo mas manipis yung kanyang cell wall, you would think na mas virulent ang gram-positive, but actually it's the other way around. because your gram-negative bacteria has the ability to excrete mga endotoxins that can actually also contribute severe reactions to their hosts, yung humans. So that's why mawawala na yung notion natin that your gram-negative are more virulent than your gram-positive. Kasi in fact, the thick peptidoglycan cell wall of your gram-positive bacteria also contributes to it more susceptible to lysozymes. So, for example, your nag-infect sila ng cell, ng host cell or animal cell, that naturally there is a bactericide in the form of your lysozymes sa mga animal cells. And they also attack the peptidoglycan cell wall. So, even if thick yung kanyang cell wall, yung protection niya, it is actually the one that is being attacked by the lysozymes. So, now, ikinaano niya pa, ikina, um, kumbaga it's a negative trait patuloy yung kanyang peptidoglycan cell wall thick. Okay? In addition, yung mga cross-linking that is found in the thick peptidoglycan is what is being attacked by your penicillins. Yan. So the penicillins na mga antibacterial agents, they would attack the cross-linking between the peptidoglycan cell wall, making your gram-negative, gram-positive bacteria now more susceptible to these penicillins. So, di ba, the thick peptidoglycan cell wall now did not really provide protection. Okay? So so presented here as well is one ano lang. So we said kasi that there is the presence of endotoxins is associated with your gram negative, but actually there is one particular gram positive bacteria, your Listeria monocytogenes, that is yan nga, gram positive siya, but actually your endotoxins are also present in your Listeria. So that's just one exception to the rule na endotoxins is just for gram negative. So also present in Listeria. Okay? So these are just a list of some gram positive and gram negative bacteria, common gram positive bacteria. So your Staphylococcus and your Streptococcus bacteria, these are gram positive. So belonging to the cocci group, the spherical in shape, we have your Staphylococcus and your Streptococcus. So under Staphylococcus, your Staphylococcus aureus, Staphylococcus epidermidis, and Saprophyticus. Tapos Streptococcus naman, you have your Streptococcus pyogenes, Streptococcus agalactiae, etc. Now, when we say Staphylococcus, yan yung clusters usually. So, dapat you would expect na yung S. aureus are clusters ang arrangement niya in your smear. Streptococcus naman, meaning chain. So, pag sinabing strepto, chain. So, dapat you would expect na when you try to visualize yung strep pyogenes or strep pneumoniae under the microscope, they would appear in chains na mga cocci. Okay? So some bacilli naman that are gram-positive, we have your Bacillus species, Clostridia species, Corynebacterium species, Listeria. Now for your gram-negative bacteria, we have for the cocci group, we have your Neisseria gonorrhoeae, the causative agent of gonorrhea, Neisseria meningitidis, Moraxella, Haemophilus influenzae. And then many of your bacteria that we have in the lab that are bacilli are actually gram-negative. So the common that we know are E. coli, Pseudomonas aeruginosa, Salmonella, Enterica, etc. Okay? So now let's review lang. So hopefully you already know this, yung mga stains that we use for gram staining. So again, we have your primary stain, your crystal violet, and then the counter stain in the form of your saffron. So ang important here to note is that we have grams iodine that acts as your mordant. So in my mind, pag sinabing mordant, so meaning it parang yung crystal violet mas mag-adhere more than before. So before, when you didn't place your crystal violet yet, sorry, your grams iodine yet, so crystal violet mo na, so it would appear violet. But when you try to place gram iodine now, so mag-increase pa yung interaction ng stain doon sa cell by forming your crystal violet iodine complex. So that's the purpose of your mordant. It increases the interaction of the stain to the cell. Now, by the time that you try to decolorize it using your 95% ethanol, um, since yung gram-positive ninyo would have your thick peptidoglycan layer plus mordant, it would be difficult to decolorize them na. So meaning even if you place your counter stain, hindi siya mag-adhere. Okay, so this was already shown to you in class. So kapag yan, so this illustration just shows us the peptidoglycan component of the cell wall. So since yung gram negative, they have a relatively thinner na peptidoglycan layer, the when it was decolorized, natanggal yung crystal violet that you placed earlier. So it became colorless. So when you applied safranin, the red or the pink na stain, it was able to absorb it na. Okay. Okay, so some common problems that are being encountered when we do gram staining. So pwedeng magkakabaliktad tuloy sila. So ano kaya yung mga possible reasons for this? Okay, or some remedies that we can do. So possible na dahil naging violet yung supposedly pink na gram negative, possible na i-decrease yung contact time ng crystal violet. Yan, so possible din na so decrease the contact time, possible masyadong matagal yung contact time kaya naging violet tuloy yung gram negative. What else? What if the other way around naman? So supposedly gram positive siya pero it appeared pink. So possible na during decolorization masyadong maraming decolorizing agent yung nailagay. So when you natanggal yung crystal violet and nung nag-counter stain, naging pink tuloy yung supposedly gram positive. Okay, so the point is sometimes due to the errors in performing the gram staining technique, there is a possibility that you actually obtain discrepancy in your results. So as much as possible, you follow the prescribed contact time, even the contact time of the mordant and the decolorizing agents, so that you will get accurate results. Okay, so next staining technique that we have is your acid-fast staining. So basically, it's a differential staining technique that you use in order to identify whether your bacteria is an acid-fast or a non-acid-fast. So generally, we have two species lang naman no, that are acid-fast, your Mycobacterium and your Nocardia species. Okay. So your acid-fast organisms, so the reason why we call them acid-fast is because they have a physical property that gives them the ability to resist decolorization during staining procedures. Yan. So actually, there is something in their cell wall or in their outer membrane that is able to retain yung stains that we give them. So that is in the form of your lipids in the lipids in the outer membrane of your bacteria. So the most common is your mycolic acids. So due to the presence of mycolic acids, the bacteria now is able to retain better yung mga stains natin even with the presence of strong decolorizers such as your acid alcohol. Okay? So again, mycolic acids, these are long chain fatty acids. So somewhat wax-like in nature. Yan. So the presence of your mycolic acids now gives you the bacteria, your mycobacterium and your nocardia, the ability to retain the stains that are placed sa kanila. Okay? So some species now that we have your nocardia, so nocardia species and mycobacterium species. So some examples are your mycobacterium tuberculosis, the causative agent of TB, of tuberculosis, and also the causative agent of leprosy, your mycobacterium leprae. So this picture just shows us, yeah, so aside from the presence of peptidoglycan in the cell wall, so sa bandang outer part, there's also the presence of your mycolic acids. That is another ano din, mga wax-like substances that contributes to the to the adherence of your stains. So yung acid fast staining, what otherwise also known as the Ziehl-Neelsen method. So that's why we call it the Ziehl-Neelsen method. It's because the method was developed by two scientists. And so when we perform yung Ziehl-Neelsen method or your acid fast staining technique, so we employ the use of two dyes similar to your gram staining. So we employ the use of a primary stain and also a counter stain. So the most common primary stain that we use is your carbol-fuchsin or your Ziehl's carbol-fuchsin. Yan. So carbol-fuchsin is red in color, so fuchsia, carbol-fuchsin. So parang fuchsia na pink. So meaning primary stain siya. So meaning kapag acid fast, it will appear blue. And then we use a counter stain. So once it is decolorized, we use a counter stain in the form of methylene blue. That's why yung mga non-acid fast, once they are decolorized, they would appear blue in color. Okay, so pakita lang natin no paano ba nangyayari. So starting with your bacteria na naturally colorless, when you apply your carbol-fuchsin, which is reddish purple in color, or the ZN stain or Ziehl-Neelsen stain, you now come up with your red bacteria. So lahat sila masustain ng carbol-fuchsin. Now, an additional step is the application of heat. So now, ang mordant natin dito is heat. So kung sa gram stain, ang mordant is grams iodine. Dito naman, you enhance the binding of the stain to the cell by applying heat. Yan. So actually, there was a different method before, yung no application of heat, known as the Kinyon method, kaya tinatawag na K-stain. Yeah. So before, you just apply yung carbol-fuchsin, tapos no mordant, but it was later on modified by Ziehl-Neelsen. They applied heat to enhance the entry of carbol-fuchsin into the cells. Yan. So by applying heat as the mordant, when you apply a very strong na decolorizing agent in the form of your acid alcohol. So since itong acid fast bacteria, they have mycolic acids that would enhance the binding of carbol-fuchsin to the cells. Yung mga non-acid fast, they do not have your mycolic acids. when you decolorize it with a strong decolorizing agent or acid alcohol, they will return to being colorless. Yan. So maging colorless again. So now when you apply when you apply your counter stain, magre-remain pink or red yung ating acid fast, but non-acid fast ones will appear the color of the counter stain, which is methylene. That's why blue. Okay. Okay. So now these are just some pictures ng itsura ng bacteria that are acid fast. So we have here yung Mycobacterium avium, wherein the red ones are your Mycobacterium species. They are the acid fast. Tapos the rest are colored blue due to methylene blue. So this one naman, it is a picture of a patient having tuberculosis. So mga bacilli that we see that are red in color, they are the Mycobacterium tuberculosis. Okay, so next we have negative staining. So when we say naman negative staining, so ano kayang stain ginagamit dito? So you would be using acidic stain. So again, when you do negative staining, you stain the background, not the cells themselves. Yan. So precaution here is that you do not fix the bacteria or the smear. You do not fix the smear in order to preserve the morphology of the bacteria. Kasi when bacterial cells are subjected to heat, they tend to shrink in size. So in order to preserve their natural morphology and you can observe them their natural state, you do not apply fixing with heat. Okay? Okay, so remember this. This is what you performed in the lab in order to prepare your smear for negative staining. Diba? You used another glass slide in order to slide yung smear and you directly inoculate your your dye. So it's not similar to what you usually do na magpa-prepare ka muna ng thin smear and fix it. So this time around, naka-inoculate talaga sa mismong dye yung bacteria. So when you push it and then you try to create a thin film, yan, that's the portion na i-observe ninyo under the microscope. So again, for negative staining, you use acidic dye. Next, we have your spore staining. So spore staining is one of our miscellaneous staining techniques wherein we try to look at or observe with emphasis yung mga spores. So again, not all bacteria are spore-forming bacteria. So there are only some types of bacteria or some species of bacteria that are able to formulate spores. And so the examples are your Bacillus species, Clostridium species, and Coxiella burnetii. So again, the purpose of your spores are it's actually for survival of your bacteria, no? So when the bacteria is subjected to unfavorable conditions, so that's when they try to form spores. Kasi diba, the spores, they are composed of outer proteinaceous na mga coating that is that are resistant to harsh environmental conditions such as yung mga extreme heat or changes in pressure and pH, for example. So nagpo-form ng spores yung bacteria, and then when the environment conditions are back to favorable conditions, so babalik sila sa vegetative state nila. Okay? So these are just some examples of pictures wherein we try to observe yung spores. So we'll look at some more pictures here. So at the same time, let's look at the differences in the spore locations when you try to when you try to report yung observation ninyo ng spore locations under the microscope. So itong picture na to would show us examples of spores that are terminally located. So meaning, pag sinabi nating terminal yung location ng spore, it is located at the far end of the spore of the bacteria. So for example, this bacteria or this one, siya ay bacilli, and you can see that yung mas matingkad na stain na part is your spores or your spores, and they are terminally located. Pag sinabi na naman natin na subterminal, so when we say subterminal, hindi siya nasa dulong-dulo, pero hindi rin siya nasa bandang gitna. So as you can see here, nandun siya sa bandang, hindi siya nasa super central, hindi rin siya nasa terminal. So it is called a subterminally located spore. Then finally, kapag naman nasa bandang center siya, so those are your centrally located na mga spores. So in this picture, those are the ones in green. And then the vegetative portions are your the pink ones. Yeah. So sometimes it can also be located outside of your bacteria. So this is just another picture showing you that the vegetative cells are stained with safranin, so pink, pero the spores are stained with malachite green, which is yung mga green na mga spores. So when we do spore staining, so yung primary stain that we use is your malachite green. So you would be staining all cells, including your spores, your endospores, your exospores, and even the vegetative cells. So the mordant that we use here is heating. So diba kasi your spores, they tend to be impermeable sa mga stains. That's why we use the aid of heating over a steam bath para mas properly mag-adhere ang malachite green on the spores. And then we decolorize it with water. So we just try to remove yung malachite green sa water. But since your spores, they were able to absorb yung malachite green, hindi siya nila ma-decolorize. They will remain green. But since na-decolorize yung vegetative cell, upon application of a counter stain in the form of your safranin, you will now stain them with pink. That's why the vegetative cells will appear pink in color when you observe them under the microscope. Okay? Okay, so lastly, the last miscellaneous staining that we have is your capsule staining. So when we say capsule staining, we want to observe the capsule portion of the bacteria. Okay, so just to give you a visualization, so this is what your bacterial cell would usually look like. So when we say capsule, so meaning it is one of the outermost part or portion of the bacterial cell, even outside of the cell wall. Okay, so we have here yung tough outer covering, which is your capsule. So the capsule is actually a portion or the outer portion of your bacteria that serves as extra protection ng mga bacterial cells. Okay? So, but sometimes another structure that is also present sa outer portion of your bacterial cells is your slime layer. And sometimes they are being interchanged or yan, interchange yung dalawang layers or dalawang structures of the bacterial cells. So, but generally, they have parang similar characteristics naman, no? So, and similar functions. So again, your capsule, it is a hydrophilic hydrophilic structure, meaning it can help in protecting the bacterial cells from drying out or desiccation. Kasi nga water siya, water loving siya, similar to your slime layer. At the same time, it also helps evade immune response of the host. Yan. And also helps in your adhering to surfaces. But the main difference of your capsule and your slime layer. So your capsule is a more organized na structure of repeating units of organic materials. On the other hand, your slime layer is a more diffused or more unorganized material. So meaning, because of the rigid characteristic of your capsule, it is not usually easily washed off um, and firmly attached to the cell surface. On the other hand, yung slime layer, it is usually easily removed with water because it is loosely attached to the surface of the cell. That's why when we observe it under the microscope, yung slime layer, we will not be able to observe that anymore kasi nga medyo loosely attached siya, easily removed siya when you prepare it sa smear, when you try to wash it off. But your capsule, that is a more easily to observe uh, na portion of the cell. Okay, your capsule lang that we can also use to refer to your capsule. So capsule, capsid, they are interchangeable terms. Ano ba ang purpose ng capsule? Why is it an important structure of your bacteria? So it actually contributes to the virulence factors of your bacteria. So the presence of a capsule around a bacteria is that um, the bacteria now is able to evade the host's immune cells or immune response. So like for example, yung human when they are infected with bacteria, because of the presence of the capsule of a bacteria, they can now evade the immune system of your host. Yeah. So it also acts as a shield against host immune recognition. So yeah. So kasi 'di ba your bacteria, yung mga bacteria that gets inside your body, usually your white blood cells will be the ones to attack that. So yung white blood cells mo, part of your immune system, would now engulf your bacteria with the process of phagocytosis. So the white blood cell is a phagocyte. It will now parang eat yung bacteria. But since the bacterial capsule helps in evading these phagocytes, hindi sila na-recognize ng phagocytes, then it is another extra protection for them. So that's the advantage of having a capsule sa bacteria. So that's the advantage no for the bacteria. But actually, in our part as developers of vaccines, we can actually use this as well, no, na to our advantage. So yung mga capsular polysaccharides or CPS, there's a trend na they can now be used as vaccine antigens. So when the capsular component of your bacteria are extracted from your bacteria, they can now be used as components of vaccines to in vaccine development. Yan. Kasi the immunogenic response ng body towards yung capsule. So, di ba, they can actually be used to um, develop your vaccines. Yan. Kasi your CPS or your capsular polysaccharides are immunogenic enough to induce protective immunity against infections of the homologous bacteria. So when we try to extract or harvest yung CPS coming from bacteria such as your H. influenzae or S. pneumoniae, Salmonella typhi, we can actually mass produce this and develop your vaccines para if the if we administer the CPS component to the human, they the human will now be able to form immunity or develop immunity against these bacteria, so that when the time comes na ma-infect sila with this bacteria, they already have um recognition of these bacteria due to the administration of your capsular polysaccharides. Okay, so that's one application in terms of vaccine development against mga bacterial infection. Yan. So in terms of performing your capsule staining technique, so remember, capsule staining is another staining technique wherein we do not perform heat fixing. Okay, so what we do is first we apply, so perform or make your smear first. Yan. So prepare your smear first, and then air dry lang siya, no? So no heat fixing. And then we apply now your primary stain, which is your crystal violet. So the purpose of it is to stain the vegetative cells. Now, we use your copper sulfate as the washing agent. So it is now used to remove your stain, your crystal violet. At the same time, it is now the stain that will come into contact with your capsule. Yan. So actually, copper sulfate is a anong tawag dito, hygroscopic na reagent. So it is very hydrophilic. It reacts with water. So since your capsular component is hydrophilic as well, yan, so it will be able to attract your copper sulfate. So now, since copper sulfate is color blue is siya, diba? So you would now expect na yung capsule, when it gets stained with copper sulfate, it will now appear bluish under the microscope. Okay. Okay. So this is what your capsule staining under the microscope would look like. So as you can see, yung vegetative cells are violet in color. Tapos the outer portion, parang there seems to be a halo that is present around the bacterial cells. So now that is your capsule. So this is actually the reason why we do not apply heat fixing, no, with your bacteria for capsule staining. Kasi again, when we heat the bacteria, there's a possibility na mag-shrink yung cells. So when the cells shrink, there appears to be a parang halo around the bacteria kasi nga nag-shrink siya. So this may come off as a parang false positive capsule. So that's why if you want to avoid a false positive result ng presence ng capsule, yan, we do not heat fix the bacteria so that the natural na size of the bacteria will remain. And if you observe halo around the bacteria, it is a true capsule talaga and not merely a space that was voided due to the shrinking of the cell when you applied heat. Okay. So some examples of bacteria that are that have capsule. So we have your Streptococcus pneumoniae, Klebsiella pneumoniae, yan. Neisseria meningitis, even your E. coli, and also your Haemophilus influenzae. Those are some examples of bacteria that have caps. Okay, so that's actually the end of our supplementary lecture. Yeah. So I hope you study well for your exam. So good luck. Thank you.